Okay, can somebody confirm you can hear me? Once again, I'm getting an error message saying my
I hear you.
speaker isn't working. Thank you. Sorry for the delay, but the verification process this time, for some reason, was really slow to verify my existence. Let's see, what are we doing today? Yep. So today is August 6th, and we should be starting chapter 6. I think we're still on chapter 5, though. All right, before we begin, are there any questions? Oh, minimize that. Any questions on anything? There was a student who requested my lecture for July 30th, and I was surprised I hadn't put that up, um, and the lab as well. Let me state that students will need to look at the July 30th lab if you want to understand the unknown project. I'm going to be sending out tonight after the lab your first results for the unknown project. You will need to begin work on the unknown project right away because it'll take you at least 2 weeks to finish that project. That's assuming you get started right away. Some students never get started on it and then get started on it right before it's due, and you will not have time to finish the project if you do that. All right, this is the last slide I have that we were talking about, and that is the integration of metabolism, even with animal cells. And animals are really bad at integrating metabolism, but we can do it. For example, when we eat a high carbohydrate diet, we don't burn all that up in aerobic respiration. We will convert a lot of those carbohydrates into lipids and then store that down. And the same thing if you were to eat a high protein diet, which we don't have that here, but put it right there, and it'll be broken down to amino acids. The amino acids will join different parts of the aerobic respiration cycle, and we will make oxaloacetic acid from certain amino acids, and then just go backwards in the Krebs cycle, and then backwards in glycolysis, making glycerol and fatty acids, and then store the excess protein down as lipids. Any question about any of that? All right, let's talk a little bit about photosynthesis. So we have about 3 or 4 more slides in this lesson before we begin chapter 6. Photosynthesis is the conversion of light energy to chemical energy. Um, Chemical energy may be used to convert atmospheric CO2 to glucose, and that we call carbon fixation, but it doesn't have to. Some of that light energy can be converted into the energy that is stored in ATP. Photosynthesis is performed by cyanobacteria, algae, and green plants. Photosynthesis is a mechanism for synthesizing organic compounds, and all organisms depend upon either photosynthesis or chemosynthesis to get their energy needs. Even animals, which do not obviously engage in photosynthesis, they must eat green plants in order to survive, and the green plants get their energy from light. There is one ecosystem which doesn't really use photosynthesis, and that's the deep sea vents and how they get their energy needs. Just as a side note, you don't need to know this, is in this ecosystem around the deep sea vent, the uh, the organisms engage in chemosynthesis, meaning it's bacteria and archaea, and they're using the energy from chemicals coming out of the deep sea vent. And then they strip the electrons from an inorganic molecule, which will be a mineral coming out of the organic vent, and get their energy needs from that way. And the other organisms in around the deep sea vent, how they get their energy needs is they eat those chemosynthetic and bacteria and archaea, or they live in symbiosis with it, like the tube worms live in symbiosis with the chemosynthetic bacteria. And then the crab, well, it will eat the tube worms. And so the whole ecosystem is running on chemosynthesis from the um, volcanic vent. But most ecosystems on the planet Earth get the majority of their energy from photosynthesis. And obviously, on land, it's mostly green plants. So photosynthesis only occurs in photosynthetic organisms. These organisms contain a light-trapping pigment. One of them is probably known to you, chlorophyll, the green pigment in plants, which make green plants green. Uh, Light actually releases an electron from chlorophyll, and then that electron is used to make chemical energy, which will be made into either ATP or glucose. And the light energy is ultimately converted into chemical energy, either ATP or glucose. And how that is done is with an electron transport chain using, once again, carrier molecules. ATP and NADPH are always generated in photosynthesis, but ATP and NADPH may be used up, meaning degraded to make glucose. And glucose will be made from CO2 and water. This process of making glucose is called carbon fixation. Any question about any of that? All right. There are two types of photosynthetic reactions, the cyclic photosynthesis and the non-cyclic photosynthesis. The non-cyclic photosynthesis is the one you probably are familiar with. And there are actually two non-cyclic photosynthetic reactions. You only need the oxygenic um, photosynthetic reaction, and that's where 6 carbon dioxide combines with 12 water molecules in the presence of light to make glucose, C6H12O6, and that generates 6 oxygen molecules, which is a waste product for the plant, and then generates 6 water molecules. You need to know this non-cyclic photosynthesis reaction. There is a non-cyclic photosynthesis reaction that does not produce oxygen, and it actually produces sulfur. And this would be in photosynthesis and an anaerobic bacteria, meaning anaerobic photosynthesis. And you don't need to know this equation. I'm just stating there is another photosynthesis reaction, which is similar. It's just not using water. It's using hydrogen sulfide gas, but it does make glucose. Like I said, this is the photosynthesis reaction that they always teach in Biology 160 or your preliminary biology course. But let me state that there is another photosynthesis reaction, and it does not make glucose. It makes ATP. And in cyclic photosynthesis, all it makes in the presence of light is light and ADP and inorganic phosphate make ATP. Very simple chemical reaction. And this occurs in cyclic photosynthesis. And you probably don't know this, but all cells that engage in photosynthesis can engage in either non-cyclic photosynthesis or cyclic photosynthesis. If the plant cell or the algae cell or the bacteria that's photosynthetic needs glucose, it'll engage in non-cyclic photosynthesis. However, if that cell needs ATP, it'll engage in cyclic photosynthesis. Any question about any of that? And obviously, ATP will be stored up for the cell's energy needs. And then whenever that cell needs energy, it will take that energy from ATP. Let's talk a little bit more about cyclic photosynthesis, also called cyclic photophosphorylation. Light excites two electrons from chlorophyll. Chlorophyll is in photosystem 1, and light hits the photosystem 1, which will be absorbed by the chlorophyll, and that will cause two electrons to leave the chlorophyll, and these electrons will travel in an electron transport chain, where the electron will move in this protein in a membrane, moving to that protein in the membrane, moving to that protein in the membrane, and then returning to this protein in the membrane. And this is an electron transport chain where we have at least 4 different proteins in a membrane. Along the way, as the electron is moving through this protein, it will give this protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. That will create a twofold gradient. There will be more hydrogen on this side than that side. There will also be more positive charges on this side than that side. By the laws of diffusion, these hydrogen ions will want to move to that side of the membrane, and they can only move through this membrane, through this protein channel. When the hydrogen ions move through this protein channel, they will give this protein the energy it needs to phosphorylate ADP to make ATP. And that's how green plants, algae, and photosynthetic bacteria make ATP in cyclic photosynthesis. Any question about that? This should be familiar to you because it's very similar to the electron transport chain of aerobic respiration. Let me see if I can get the graph here. There it is. So here's the electron transport chain of aerobic respiration. Let me blow that up so you can see it a little better. And the electrons are moving through the proteins in a membrane, and they're giving some of the proteins the energy they need to move the hydrogen ions from this side of the membrane to that side of the membrane. That hydrogen ions creates potential energy, and that potential energy will be used to make ATP. The hydrogen ions can only move through this channel protein, which is an ATP synthase protein. And when the hydrogen ions move through this protein, they give the protein the energy it needs to phosphorylate ADP to make ATP. Any question about any of that? A little further. Why is this called cyclic photosynthesis? Because the electron starts in photosystem 1 and the chlorophyll is in, come on, mouse. The electron is in photosystem 1 in the chlorophyll. The electron moves to this protein, moves to that protein, moves to that protein, and then returns to its starting position, which is the chlorophyll. Why it's called cyclic photosynthesis, because the electron moves in a circle, a cycle. Any question about any of that? All right, so that's how in cyclic photosynthesis, the photosynthetic cell can make ATP. So the energy source can vary for making ATP. It can come from the sun in a photosynthetic organism. It can also come from glucose or some elemental non, what am I trying to say here? An elemental element, like sulfur or ammonia or hydrogen gas, some chemical, that's what I'm looking for, where the electrons will be stripped off of the sulfur or the ammonia or hydrogen sulfide gas. It's very similar to stripping off the electrons from glucose. And then those electrons are used to make ATP by largely the electrons we carried by an electron carrier molecule, which we talked about, NAD plus and FAD in aerobic respiration. You don't need to know NADP plus, but that's the electron carrier in photosynthetic organisms. And then the final electron acceptor can vary. In aerobic respiration, the final electron acceptor will be oxygen. That's where the electrons from glucose or sulfur will end up in aerobic respiration. There's also anaerobic respiration where the final electron acceptor will be a molecule other than oxygen, and that'll happen in anaerobic respiration. And that can be different chemical agents like nitrate or sulfate, just depending on which form of anaerobic respiration we're talking about. And there's different forms which use different molecules. I mean, there's many different, there's iron and there's hydrogen sulfide gas, and there's a whole bunch of ones. And then in fermentation, we use as the final electron acceptor an organic compound like pyruvate, which will be converted into its fermentation end product, which for lactic acid fermentation will be lactic acid, also called lactate. It's not shown here, but the final electron acceptor in cyclic photosynthesis is chlorophyll. Any question about any of that? All right, so the point is the energy source can vary and the final electron acceptor can also vary. Any questions about chapter 5? If not, let's begin chapter 6. Let me save that down. As is normal, I show you the slide saying what it is. Chapter 6, microbial growth. I introduce the major terms. You should know all the terms. In fact, you should know all the objectives, which you can get from the study guide for chapter 6 on my webpage. But the major goals are for the terms psychrophiles, psychotrophs, mesophiles, thermophiles. Know what those are? Hypothermophiles, acetophiles, halophiles. There's an aerotolerant anaerobe, a facultative anaerobe, a binary fission. Know what a reproductive spore is and how it differs from an endospore. And then 2, understand the various requirements of cells for growth. 3, understand the 4 phases of growth. And then 4, compare the different methods we have for measuring microbial growth. Any questions about this overview and summary? All right. When we're talking about microbial growth, you should know that I'm referring to an increase in the number of cells, not an increase in the cell size. And here we're going to look at a little video about microbial growth.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
All right, any question about that? And so when we're talking about microbes, when we're referring to growth, we mean something different than the increase in size of the body. I used to teach middle school students, and when I meant about them growing, obviously I was not talking about them increasing in number. All right, there are different requirements for microbial growth. There are physical requirements and chemical requirements. Some physical requirements: all organisms need to be at the correct temperature, pH, osmotic pressure, or salinity, and etc. Meaning to engage in growth, you must be in the correct environment. You also need various chemical sources in order to have microbial growth. All cells require a source of carbon, a source of nitrogen, a source of sulfur, a source of phosphorus, or source of trace elements, a source of oxygen if they require oxygen, or the absence of oxygen if they require the absence of oxygen. And if cells require organic growth factors, that must be present in order for the cells to grow. Any question about any of that? Does anyone know an organic growth factor that humans have? I'll give you a hint. An organic growth factor is a biological molecule. So what's a biological molecule that humans must require? Anyone? Nobody has any idea what an organic growth factor is?
Carbohydrates.
No, that would be a source of energy as well as carbon for us, but that's a good guess. No, a vitamin would be an organic growth factor, like vitamin C. We must have it because we can't make it. And if it's not in our diet, we will become sick and eventually we will die, meaning we do require vitamin C as well as all vitamins. And that's why it's called an organic growth factor. There are a few other things besides vitamin, like essential amino acids, essential fatty acids. These are either amino acids or fatty acids that our body cannot make, and so we must get them in our diet. An example of an essential amino acid would be the amino acid lysine. We must get it in our diet, or else we'd become sick and eventually die, because we cannot make that amino acid. And those are what organic growth factors are. Each species has its own minimum, maximum, and optimum growth temperature. The minimum would be the minimum temperature the organism can grow at, and if you drop the temperature below the minimum, the organism cannot grow. The same with maximum. If you raise the temperature any higher, the organism cannot grow. And then optimum is the temperature that the organism grows fastest at. Any question about that? So like E. coli would grow fastest at 37 degrees, and that's why E. coli likes to be in people's guts. It can grow like on an apple or even in the soil, but that would not be its optimum growth temperature. Any question about that? All right, microorganisms classified into groups on the basis of their preferred temperature range. We have psychrophiles, and these organisms grow best around 10 degrees Celsius or a little lower. a little higher, around 10 degrees, we'll just say that. And these would be organisms that grow in your refrigerator. Why you should not keep foods that don't have a preservative in it in your refrigerator for longer than a week is that the psychrophiles can grow to significant numbers in a week, and after a week could cause spoilage of the food. So food like meat and I don't know, beans and anything that's soft and doesn't have preservatives in it, you put it in the refrigerator. After about a week, you should throw it away if you haven't eaten it because the psychrophiles can grow in the refrigerator. The psychotrophs are organisms that grow a little bit above a psychrophile, and these are like organisms that grow best around room temperature. The mesophiles are organisms that grow best around 37 degrees. All human pathogens are mesophiles because these organisms grow best at around our body temperature. Any question about that? And then we have thermophiles. These are organisms that grow best around 60 degrees Celsius. Whenever you see a chicken under a heat lamp in the store, like at Safeway, and it's going around and around and around, and you look at that and you say, geez, that chicken looks pretty old. It's been sitting there for a long time. And the thermophiles can actually grow under that heat lamp. And so if that chicken looks really old, you might not want to consider buying it because the thermophiles can grow in that chicken. It's in the under the heat lamp. Hypothermophiles were something that was not known when I took microbiology. These are organisms that grow best around 95 degrees Celsius. Recall that 100 degrees Celsius is the temperature of boiling water. And these organisms grow best around 95 degrees Celsius. Now understand that the temperature ranges as well as the optimum is not strictly defined. Like a mesophile is something that grows best under this graph around 37 degrees. But when we have Staphylococcus epidermidis, which grows on your skin, its optimum temperature growth is actually around 30 degrees because our skin is a little cooler than inside our body. And so it's closer to around 30 or 32 degrees. And that would be the optimum for Staphylococcus epidermidis to grow because it colonizes the skin. And like I said, its optimum highest, fastest, it'll grow best around 30 degrees. It can grow at 37 degrees, but that's not the optimum. Okay, so my point is that this is not strictly defined. Anything that's around 37 degrees would consider a mesophile. And then the range especially varies. Like with the psychotrophs, this graph shows a minus 10 degrees up to about 20 degrees. Well, as you know, there are some organisms that make, oh, what is that called? An antifreeze. And so I think the coldest cell can actually survive something down to a minus 20 degrees Celsius because of all the antifreeze in the cell. And so that cell could grow down to a minus 20 degrees. Below that, the cell will freeze and it may not die, especially if it's a prokaryote, but it cannot grow because when it's frozen, it cannot engage in metabolism. But once it thaws out and the temperature raises, that cell could continue growing. Any question about any of that? So different organisms have different optimums. like Staphylococcus epidermidis, Micrococcus luteus, their optimum is 30 degrees. I think Staphylococcus aureus, maybe not though. Let me think about that. Let's say Streptococcus pyogenes, which grows in the throat. Its optimum is around, or inside the body, is around 37 degrees. All right, any question about any of that? Let's talk a little bit about food spoilage. Let me blow this up. Food that is between 60 degrees Fahrenheit to about 130 degrees Fahrenheit, and that would be about 16 degrees Celsius to 51 or 52 degrees Celsius. That food is in the danger zone. We have rapid growth of bacteria. Some of them may grow and then produce toxins. Food between 60 and 130 degrees Fahrenheit should not be kept at this temperature for very long, like 4 hours or more. After 4 hours, that food should be thrown away because the bacteria can rapidly grow. Now, bacteria can grow a little bit slower between, I don't know, 150 degrees Fahrenheit down to about, it looks like about maybe 37 degrees Fahrenheit. So bacteria can grow in an extended range, but this growth will be much slower. So any food that's kept at this temperature for any length of time should be thrown away. Food spoilage can happen because psychotroph and mesophiles can grow at this temperature and cause the food to spoil, especially if they produce a toxin, in which case the food will look good and will taste good. The toxin won't produce a taste, but then when you eat it, you will get sick from the toxin. Food, when it's refrigerated, should be put in a small vat so that the food uh, cools below 15 degrees within something like, let's see, a half an hour or an hour. Any food that's in too large of a vat and then is refrigerated will spend several hours. How many hours is that? 1, 2, 3, 4, 5 hours or more in the danger zone, in which case the food will not cool fast enough and the microorganisms can grow in this temperature and then can cause the food to spoil. So don't ever refrigerate something in a very large vat because the food will not cool fast enough. It needs to move through this temperature range in an hour or less. Most bacteria grow best between pH around pH 7, so something like pH 6 to 8 or 6.5 to 7.5. Yeah, something around pH 7. Lab cultures. This is a problem for lab cultures because the bacteria tend to grow and then make acidic microbial products, which can then inhibit the growth of the microbe. We tend to minimize this in a lab culture, meaning if you get a tube of media from the lab and then grow your culture in it, we use pH buffers, put them in the buffer or the media to reduce the rapid change in the pH from the microbes. And that way the microbes can grow longer because the pH is buffered. It'll stay around pH 7 for a longer period of time. Molds and yeast grow in slightly acidic environments, and their optimum is actually down to pH 5 or pH 6. And so they can grow better, a little bit more acidic environment. Now, bacteria can grow in acid environments. These bacteria we call acidophiles, bacteria or microbes that can grow in acid environments. The most acidic acidophiles can actually grow in a volcanic environment where the pH can be around pH 0 or between pH 0 to pH 3. If you remember, this pH of your stomach is around pH 2. It could be as low as pH 1.5. And we do have microbes that grow in that. Helicobacter pylori is actually a microbe which likes acidic environments, and we can find that microbe growing in your stomach. Any question about any of that? All right. Cells have physical requirements for growth. One is osmotic pressure. Microbes require a certain amount of water in order to grow. If the cell is under high osmotic pressure, this will remove the water necessary from a cell, so the cell cannot grow. And in fact, the cell, depending on what type of cell it is, could die in from high osmotic pressure. A hypertonic environment where the solute concentration is higher outside the cell than inside the cell, such as an environment with high salt or high sugar, will prevent the cell's growth. Like if E. coli falls in molasses, the water will leave the cell membrane will shrink, pull away from the cell wall, and then the cell, especially if it's E. coli, will die by plasmolysis. Plasmolysis always inhibits growth, and it may kill the cell depending on how sensitive the cell is to desiccation, like E. coli would not survive for very long in a bottle of molasses or wherever there's hypertonic environment. But something like Staphylococcus aureus can survive for a longer period of time. It will not grow in a bottle of molasses, but it can survive for a longer period of time than E. coli. Eventually, though, it will die. So when Staphylococcus aureus does fall in the bottle of molasses, it can survive for a while, but eventually it will die. And that's why the bottle of molasses can stay in your cupboard for room temperature for over a year and it's still good. My mother had a bottle of molasses that we figured was over 30 years old, and it was still good, although it hadn't been used for 10 years, meaning it was 20 years old when I used it to make some molasses candy for my father, and he enjoyed it, didn't kill him or anything like that. And we never used it for 10 years after that. So my mother said, I think it's time to throw this bottle away. And I said, you may as well because I don't use it and you're not using it. So we threw it away. Anyways, high osmotic pressure can be used to preserve foods. And we do that by salting meat, molasses, salted fish. Any question about any of that? Halophiles are organisms that prefer living in salt. Obligate halophiles require a high salt concentration to grow. A facultative halophile do not require the high salt concentration to grow, but they can tolerate salt greater than 2%. Some can grow in salt up to 15% salt. So an example of a facultative halophile would be Staphylococcus epidermidis or Staphylococcus aureus. It can grow on your skin, which actually gets up to 20% salt, sodium chloride. And it doesn't require that salt concentration to grow. It can grow on a TSA plate, which actually only has about 0.9% sodium chloride, but it can grow on your skin, which the very highest it can get would be 20%. Obviously, it would depend on how much you've been sweating and when you last took your shower. But your salt can get up to 20% on your skin. In a hypotonic solution, that's low osmotic pressure, where there's lots of distilled water around, water will enter the cell by osmosis. And the cell will be lysed if the cell does not have a cell wall. Put your red blood cell in a beaker of water, water will enter the cell, causing the cell to puff, and the cell membrane will expand until it pops, and then the cell will die. But if the cell has a cell wall, like most bacteria, as well as Euglena, trying to think of what that is called. An algae has a cell wall, and it can be in water, and the water will enter the cell up until the cell membrane touches the cell wall. After that point, the cell membrane cannot expand any further because of the cell wall, and that will prevent any further water from moving into the cell simply because there is no space for the water, meaning the cell membrane cannot expand, and so that will prevent the cell from dying or undergoing rupture of the cell membrane because the cell wall protects the cell membrane. Any question about that? All right. One chemical requirement that all cells have is a source of carbon. All cells must have a source of carbon in order to survive and grow. Carbon typically is about half of the weight of the dry weight of cells. So about half of the weight, once the cell has lost its water, meaning the dry weight, will be carbon. Carbon is used in structural organic molecules. It is used as an energy source for the cell. Heterotrophs use an organic carbon source, like we are a heterotroph, and for us to get carbon, we must consume it, such as glucose or any other food source, but we must get carbon as a carbon source, and we're a little bit different. The same carbon source will also be our same energy source, and we get most of our energy as well as most of our carbon from glucose or some food molecule. An autotroph, though, can get its carbon from carbon dioxide, either dissolved in the water or in the air. So an autotroph can actually get its carbon source from CO2. Nitrogen, sulfur, and phosphorus, three other chemical requirements. Nitrogen is actually 14% of the dry weight of a cell. You don't really need to know that, but nitrogen is in amino acids, proteins, DNA, RNA, and ATP. Nitrogen is often obtained by decomposition of proteins, and that's where humans get most of our nitrogen from. Many bacteria, though, can get nitrogen from either ammonium or nitrate or some other nitrogen source in the soil or water. We cannot get our nitrogen from ammonium or nitrate, but green plants can. In fact, green plants can use both of these. They prefer nitrate. Okay. A few bacteria can even get nitrogen from nitrogen gas in the air, and these we call the bacteria or archaea that can engage in nitrogen fixation. They can bring down nitrogen from the air and then put it in the soil. Sulfur, another chemical requirement. It's in amino acids, in the B vitamins, thiamine, and biotin. This is required for DNA and RNA synthesis. Most organisms obtain their sulfur from decomposition of proteins, but some bacteria can use sulfate. Actually, many bacteria can use sulfate. Some can use hydrogen sulfide as a sulfur source. We cannot. In fact, hydrogen sulfide is toxic to us. Green plants can use sulfate. Phosphorus, it's required for DNA, RNA, and ATP production. It's also required to making membranes, meaning we need phosphorus to make phospholipids, which is making the membranes. Phosphate is an important source of phosphorus, and we can get phosphates from the food we eat and the liquids we drink, like you can get phosphate from milk, for example. And yes, we can use phosphate, although I think we get a lot of our phosphorus from eating phospholipids, like when we eat a cell and it has a membrane. Any question about that? So most bacteria can get phosphorus from phosphate. Trace elements. These are organic elements required by microbes in order to grow, but they only require them in very small amounts. They are often required as an enzyme cofactor. Some examples of a trace element would be iron, copper, and zinc. And humans do require trace elements too, but usually trace elements are picked up from the environment for a bacteria cell, but would be in the food we eat or the liquids we drink. Like we get iron from certain vegetables, we get it from red meat. Copper can be obtained from raisins, obviously from other fruit, I believe, as well as probably some vegetables. You can get some copper, not a lot, but you can get some copper from meat. And I don't know about the source of zinc, where we get zinc from, so I'm not going to try and make that one up. But uh, these are often just dissolved in the water or the liquid we drink or the food we consume. One thing in Western Washington and Western Oregon that is a trace element, which we cannot get from our environment, is iodine. This is required by humans as a trace element. And because of all the rain in Western Washington and Western Oregon, it's washed iodine out of the soil. And so if we were to only eat local plants and animals, we would not get enough iodine. However, they put iodine in iodized salt. And then, of course, if you get food grown in another location in the world, which could be many places like Eastern Washington or Colorado, you'll get iodine in the food grown in those locations. Any question about any of that?
No question, but you can get zinc from like pumpkin seeds and sesame seeds.
Okay, great. Thank you. Did not know that, but now I do. All right. Organic growth factors are also chemical requirement. These are organic compounds obtained from the environment that the organism cannot synthesize itself. Examples include vitamins, certain amino acids, and certain essential fatty acids. They're called, for humans, essential fatty acids and essential amino acid. For bacteria, it could be certain purines and pyrimidines. If they cannot make it, they must get it from their environment. And usually, if they require a purine or a pyrimidine, it may be a pathogen. And where that pathogen gets its purine or pyrimidine would be from killing your cell and then getting the purines and pyrimidines from your cells. So that would be from obtaining it from DNA or RNA, breaking it down and getting the purines and pyrimidines from either DNA or RNA. So vitamin C would be an organic growth factor required by humans because we must get it from our diet. But mice and bacteria do not require vitamin C because if they need it, like the mouse does, but the mouse can synthesize its own vitamin C. All right, oxygen is a chemical requirement for many organisms, but molecular oxygen is, in a sense, a toxic substance. Even for aerobes, some intermediate forms of oxygen are toxic. And there are 4 toxic forms of oxygen that our cells, as well as any aerobe, as well as any anaerobe, must remove, or the toxic form of oxygen will kill the cell. There's singlet oxygen. This would be O2 boosted to a high energy state where the electron is highly reactive. And actually, we can find singlet oxygen in our white blood cells, phagocytic white blood cells. And then the white blood cell can release singlet oxygen on something it's trying to kill. So we must remove singlet oxygen, otherwise it will be toxic to us, except for obviously our phagocytic white blood cells. And what they do is they keep that in a membrane-bound organelle, the singlet oxygen. I'm trying to remember what that's called, the membrane-bound organelle lysozyme. Is at the end, lysosome, a lysosome. So singlet oxygen can be in a lysosome of a phagocytic white blood cell. And we have enzymes for removing singlet oxygen. Superoxide free radical. This is oxygen with an extra electron. That symbol refers to an unpaired electron, and that symbol is, or the unpaired electron, is only found in free radicals. Free radicals are highly unstable. Whatever they bump into, they tend to steal an electron from whatever they bump into. Superoxide free radicals are produced in normal aerobic respiration when oxygen is used as the final electron acceptor. And then they'll bump into something and steal an electron from it, which could steal the electron from the neighboring molecule, making that molecule a free radical. And then it can bump into another molecule, which can steal the electron from that molecule. And the danger is it can mutate DNA as well as damage whatever that molecule is. And so we need to neutralize superoxide free radical. And how we do this is we have an enzyme, superoxide dismutase, or SOD, which can convert the superoxide free radical right here into hydrogen peroxide. Hydrogen peroxide is also toxic to the cell, but that's our third toxic form of an oxygen. Hydrogen peroxide is a peroxide anion. It's toxic to cells. And 2 peroxide anions can combine with 2 hydrogen ions to form oxygen and hydrogen peroxide, which is toxic. It's a peroxide ion. We have 2 enzymes to catabolize hydrogen peroxide. There's the enzyme catalase, which can take 2 hydrogen peroxide to make 2 water and oxygen. And then there's also the enzyme peroxidase, which can take hydrogen peroxide and 2 hydrogens to make 2 waters. Catalase is the most important enzyme in our cells to remove hydrogen peroxide. And so if you ever put hydrogen peroxide on your skin, you can feel the bubbling, and that's from the generation of oxygen making those bubbles. Bacteria that can grow in air, and if the bacteria do not have the enzyme catalase and they can grow in air, that means they have the enzyme peroxidase. And now cells can remove hydrogen peroxide and make water. Any question about that? All right. So the fourth toxic form of oxygen is simply a hydroxyl radical. This is hydroxide with an extra electron, but it's an unpaired electron. And then this hydroxyl radical can bump into something and steal an electron from it. Remember that this hydroxyl radical differs from hydroxide ion, which has actually 2 extra electrons and a negative charge of 1, or the hydroxyl group in alcohol, meaning this would be ROH. These are not a hydroxyl radical when OH group or the hydroxide ion bump into molecules. They do not steal an electron from them. Only the hydroxyl radical steals an electron from something. Hydroxyl radicals are made by aerobic respiration in trace amounts. It can be made by ionizing radiation, or when hydrogen peroxide reacts with a certain metal ion, it can produce a hydroxyl radical. Our cells have antioxidants to remove hydroxyl radicals and other free radicals. In one example of an antioxidant is vitamin C and vitamin E. They can combine with the hydroxyl radical and then eliminate it. That does use up the vitamin C or the vitamin E, but you don't want hydroxyl radicals around. And like I said, we have antioxidants to remove hydroxyl radicals. Any question about any of that? All right, so those are the 4 toxic forms of oxygen. All organisms must remove the toxic forms of oxygen if oxygen is around, and these are all intermediates of oxygen metabolism, or these toxic forms of oxygen will kill the cell. Facultative anaerobes will only grow in the absence of oxygen because oxygen, particularly one of the 4 toxic forms of oxygen, will kill that cell. And the anaerobe, which is what am I trying to say here? A facultative anaerobe does not have the enzymes to remove the toxic forms of oxygen. So those cells must grow in an oxygen depleted environment. Microbes that use oxygen we call aerobes. They do produce more ATP from nutrients than anaerobes, organisms that do not use oxygen. And therefore aerobes grow faster than anaerobes, or tend to grow faster than anaerobes. Microbes are classified on the basis of the effect oxygen has on their growth. And we have 5 different classifications depending on the effect oxygen has on their growth. First, there's the aerobes. These are organisms that grow in the presence of oxygen, and they require oxygen around in order to grow. And that's because they engage in aerobic respiration, and that's the only way they can make energy to grow. These organisms grow at the top of a, let me see what the name of this is called. Forgetting the name of this media. Oh, it'll come to me. This media has it has a chemical in it, which I'm not remembering the name of because I can't remember the name of the media, but it has a chemical in it which combines with oxygen to remove the oxygen. And when you first take the tube out of the autoclave, there will be no oxygen in the media, but there's oxygen in the air. And so the top of the tube will be full oxygen, 20 to 21% oxygen. And as you go down the tube, there will be less oxygen. Until you get to the bottom of the tube, there will be no oxygen. What's the name of that? Sorry, I'm not remembering the name of this media. Oh, it'll come to me. Just, I'll have to give it to you later. Anyways, an obligate aerobe, organisms that can only grow in the presence of oxygen, will grow best at the very top of the tube. That's why there's so many organisms at the top. And as they grow down the tube, there should be one right there, there will be less and less growth because as you go down the tube, there's less and less oxygen. And then at the bottom of the tube, there will be no growth because there is no oxygen in the bottom of the tube. So the organisms cannot grow. Obviously, we are an aerobe and we must grow in the presence of oxygen. In fact, we don't do very good in reduced oxygen, but microbes can have slower growth at lower oxygen concentrations. So like I said, there'd be like one cell here capable of growing. So there should be one cell right there. That differs from an obligate anaerobe. These organisms only grow in the absence of oxygen, and they will have most growth at the bottom of the tube where there is no oxygen. As you go up the tube, there will be more and more oxygen, and so there will be less and less growth. So that's why there's less growth here, and there should be one right here because there is a little bit of growth and a little bit of oxygen because cells can grow where there's no oxygen. There's only a little bit of oxygen. So they'll have less and less growth the further up you go in the tube because there will be more and more oxygen. And obviously at the top of the tube, there will be no growth. And that's an obligate anaerobe. These organisms have no enzymes or metabolites or antioxidants to remove the toxic oxygen forms. That differs from a facultative anaerobe, which can engage in aerobic respiration, and it can engage in anaerobic respiration or fermentation. And in a facultative anaerobe, we'll see the best and highest growth at the very top of the tube. That's because the cells can grow fastest here in full oxygen because they're engaging in aerobic respiration. But they can use anaerobic respiration or fermentation to grow. And so we can see growth throughout the tube. And as you go down the tube, you'll see less and less growth. So at the bottom of the tube, we only have one cell. And that's because they can grow slowly in the absence of oxygen, either from anaerobic respiration or fermentation. But as you grow up the tube, you'll see more and more growth. The highest growth at the very top of the tube, where the cells can go engage in aerobic respiration. And they grow the best in full oxygen because they can make the most ATP engaging in aerobic respiration. So facultative anaerobes have growth throughout the tube, highest growth at the very top of the tube. An aerotolerant anaerobe is an anaerobe which does not use oxygen, why it's called an anaerobe, but it has all of the enzymes and antioxidants to remove the toxic forms of oxygen. These anaerobes can grow anywhere in the tube, and they have uniform growth at the top of the tube and the bottom of the tube because these organisms do not need oxygen to grow, and they have all the enzymes and molecules they need to remove the toxic oxygen molecules or the four toxic forms of oxygen. And so they can grow uniformly throughout the tube. There are not too many organisms that are aerotolerant anaerobes, but there are some. And then there's a microaerophile. These organisms are aerobes, so they do require some oxygen to grow. They will not grow at the bottom of the tube because they do require some oxygen to grow. They are an aerobe. However, they do not grow best at full oxygen. So at the top of the tube, come on, mouse, there's either no growth or very little growth. And at the bottom of the tube, there's no growth. And they grow best in the middle of the tube where the oxygen level is reduced, usually between 2 to 10%. And when you move away from that, they will have some growth, like one dot here and one dot there. They'll have some growth when you move away from the middle of the tube, but they'll have less growth than in the middle of the tube. So they require oxygen to grow, but they grow best at reduced oxygen concentrations. And one example of an organism that is that way is Helobacter pylori. It is a microaerophile. It grows best in the stomach, which has between usually 2 to 4% oxygen. And the reason why the oxygen is reduced in the stomach is that the sphincter is closed, so there's no oxygen coming from the mouth or the air. And what oxygen which arrives in the stomach, there's microbes in the stomach which will use up that oxygen, reducing the oxygen in the stomach to between usually 2 to 4%. Obviously, if you were to swallow a whole bunch of times, you would put oxygen in the stomach and it would increase, but there are microbes which will reduce that oxygen. All right, have I talked about all of these now? Yes, I have. All right. Thioglycolate. That's what's in this media. These are called thioglycolate tubes, and they have thioglycolate as a chemical added to the media, and thioglycolate will combine with oxygen and then remove it. So at the bottom of the tube, there's no oxygen. The top of the tube, there's full oxygen, and thioglycolate is used up at the top of the tube. Any questions about that? See, I didn't get the name of that. It just took me a while to get it. Thioglycolate tubes. Let's talk a little bit about culture media. I don't know if I mentioned it. Let me get out something here. We have in microbiology a book in the lab that is actually a little bit bigger than this, but it's about this thick. So we have a book in the lab, and on each page of this book, we have a media. So each page is discussing how to make a certain media, and then it might talk a little bit about what you use the media for. The point is, in microbiology, we have a large number of different media, because obviously each page of this book discusses one media. And the book is really thick, as well as fairly big. When we're talking about media, media is a word for more than one, it's plural. Whenever we're talking about one medium, you should use the word medium because that's the single word for media, the plural. It is a liquid or a solid solution which provides all of the nutrients required by organisms to grow. And if you were to come into a microbiology lab, we would supply a tube or a plate of media for you guys to grow your microbes. Obviously, we're going to be doing this online, and so you just need to know that there's different types of culture media, and that when we supply them to you in the lab, it'll be sterile, and that is a tube or a plate that has no living microbes in it. That may not refer to let me think about what the heck that's called. Not doing too well recalling certain words today. It may not have a prion. The media we supply may not have any living microbes, but it could have a prion in it. Unlikely to, because prions wouldn't be normally around in a media, but it may not refer to viruses either. They could have viruses in the media, although most viruses would be killed by autoclaving, so there probably wouldn't be viruses. But when we say sterile, we mean it has no living microbes, meaning no living cells in it. Inoculum is the microbes we introduce into the media. So when you introduce microbes into a tube of media, that would be the inoculum. The culture is the microbes growing in and on a culture medium. And like I said, we have a very wide variety of media available in microbiology. We won't discuss, obviously, the entire book. We're only going to discuss a few types of media. So media support microbial growth. A medium must provide an energy source, a carbon source, a source of nitrogen, a source of sulfur, a source of phosphorus, trace elements, and organic growth factors if an organism needs them. Like E. coli doesn't need any growth factors, but it does require trace elements, does require nitrogen, sulfur, phosphorus source, a carbon source, and an energy source. If you were to grow a autotrophic microbe and you grow it in the light, you do not need an energy source because the light will provide the energy. You do not need a carbon source because it's an autotroph. It can get carbon from carbon dioxide in the air or dissolve in the media, liquid media. You may need a nitrogen source. The exception would be for the autotroph that can actually fix nitrogen from the air. You would need a sulfur source. You wouldn't need a phosphorus source. You would need trace elements, but you can get the trace elements usually just by using tap water. And a autotroph that you're growing would not need an organic growth factor. Any questions about any of that? So it depends really what you're growing, depending on what you need to supply. And usually we grow things like E. coli, which would need an energy source, a carbon source, a source of nitrogen, sulfur, phosphorus, would not need trace elements if we're using tap water, and would not need growth factors, although it does require trace elements, but the tap water would supply it. Agar is something we add to solidify a liquid media to make a solid media. It is a complex polysaccharide derived from seaweed. We usually use agar when we're talking about Petri plates because a liquid in a Petri dish would be just too easy to spill out. So we usually use plates with agar in them, and then you're unlikely to spill the agar out. We can also use agar in a tube. We have two types, a slant and a deep. Let me show you. There it is. So if we have a tube like this, and the agar media is simply across like that, that's called the deep. So down here is the agar. And then the slant would be something like where we have the media growing in a slant like that. And then you grow the bacteria usually on the slant there. Any question about any of that? So a deep right here, and then a slant. Come on, mouse. On the right here, I'm not getting my mouse to show up. I don't know why. Oh, there it is. Well, there's now disappeared again. Agar is generally not metabolized by microbes and why it's commonly used to solidify a liquid media. You could, excuse me, you could use either starch or gelatin to also solidify the liquid media and make it a solid. But some microbes can metabolize starch or gelatin. And so we do use them, but not as much as we use agar. Any question about any of that? And usually when we're using starch, we're just looking to see if the organism can metabolize the starch. And often when we use gelatin, we're looking to see if the organism can metabolize the gelatin as well. As I stated, we have different types of culture media. We have chemically defined media, and these are media where we know the exact chemical composition of the media because every molecule we've added, we specifically added ourselves. So we know that what it is. Let me blow that up a little bit. One chemically defined media, this is, I think, glucose media. The main ingredient is 5 grams of glucose, which is both a carbon and an energy source. We do provide ammonium phosphate, monobasic, to provide a source of nitrogen as well as a source of phosphorus. We do include sodium chloride to keep the ion content. What am I trying to say here? The media that the correct osmolality, meaning the correct isotonic media. We do include magnesium sulfate, both as a source of magnesium as well as a source of sulfate or sulfur. We do include potassium phosphate to provide potassium as well as phosphate. And then we have water. And that's all the molecules that are in this media. So that's a chemically defined media. We know exactly what is in this media. Now, many organisms cannot grow in this media. And that would be an organism that would be fastidious, in which case you must supply specific molecules in order for the organism to grow. And a fastidious organism is one that does not grow easily. It's an organism where certain minerals, chemicals, or nutrients must be supplied in order for the organism to grow. And this would be a chemically defined media for a fastidious organism, which is obviously having many more stuff added. These are kind of normal. That's pretty normal. But this is providing a carbon source. This is providing a sulfur source. More sulfur. I'm not sure why there's two different sulfur sources. Magnesium source, ammonia or nitrogen source, a potassium source, a calcium source, an iron source, as well as a nitrogen source. A source of cysteine, that's an amino acid, a source of arginine, glutamic acid, asparagine, cysteine. Why do we have two cysteines in there? Oh, cysteine, sorry. Two different amino acids. These are all amino acids that are supplied. And then organic growth factors. Not even going to read through them. These two are B vitamins. I think biotin's a B vitamin too. And then this, we have a reducing agent, sodium thioglycolate, that will be to remove oxygen. So this would be added for an anaerobe, and then you grow the organism at the bottom of the tube. The point is, this is a chemically defined organism for a fastidious organism, such as growing Neisseria gonorrhoeae, where you must supply many different chemicals in order for the organism to grow. A chemically defined media is usually used only for growing an autotroph because they are easy to grow and you don't need to supply many things for an autotroph to grow. Although I think E. coli would actually grow in this glucose salts media right here. For many organisms, we grow them in a complex media where the exact chemical makeup of the media is not known. And that's because we add extracts of digestive yeast, meat, or plants. And like if you're adding, let's just blow that up so I can show it better. This is a chemically complex media where we're adding beef extract, 3 grams of it, and we will be adding all the molecules that are in the cows, because this is probably from more than one cow. And whatever molecule the cow had been eating will be in this beef extract. And obviously that would depend on where the cow is and what plants it's eating. So the extracts of yeast, meats, or plants provide vitamins or organic growth factors, but the exact chemical composition is not known, and it will change too from the different vat to the different vat. Like if this uses one cow from Colorado, and the next bottle we open has a cow from, I don't know, Georgia, they will contain different molecules. Peptone is a partial protein digest. It's often added to these complex medias. And usually we get this protein digest from soybeans. But it adds a primary energy source, a carbon source, a nitrogen source, and a sulfur source, much as the meat does. The meat adds a primary energy source, a carbon source, a nitrogen source, a sulfur source. The meat also adds a phosphorus source. We use these complex medias to grow most heterotrophs, bacteria and fungi that require an organic carbon source. Any question about any of that? Two complex media that we frequently use in microbiology would be tryptic soy agar, or TSA, and nutrient agar. And this right here is actually nutrient agar. We do have other types of media. We have an anaerobic media, and this will be an important media for growing obligate anaerobes, organisms that must avoid the exposure to oxygen. An anaerobic media is sometimes called the reducing media because these media typically have chemicals in it, like sodium thioglycolate, that reduce the oxygen in the media and combine with and remove the oxygen, resulting in oxygen depletion. And as I mentioned, in a thioglycolate tube, at the bottom of the tube, there will be no oxygen, and we can grow a facultative anaerobe in the bottom of the tube of the thioglycolate tube. What you do is you heat the media. For one thing, you want to sterilize it in an autoclave. That will remove the absorbed oxygen in the media. And then at the bottom of the tube, there will be no oxygen because the thioglycolate will remove any oxygen. But at the top of the tube, oxygen will be coming in, and the sodium thioglycolate will be used all up at the top of the tube. And that's why a thioglycolate tube has full oxygen at the top of the tube and then no oxygen at the bottom of the tube. And in the middle of the tube, it'll be reduced oxygen the further down you go in the tube. Any question about any of that? Oh, come on. Sorry, I clicked that by mistake. All right. We also have a selective media. This is a media that has a chemical in it that halts or inhibits the growth of a particular group of microorganisms while not affecting the growth of another type of organism. One example of a selective media is 7% sodium chloride media. This media is made up of 7% sodium chloride, which is way too much salt for most organisms to grow. Only organisms that like salt can grow in 7% sodium chloride media. So most organisms cannot grow above 2% sodium chloride, but obviously a halophile, an organism that likes salt, can grow on 7% sodium chloride media. Halophile means salt lover, by the way. We also have a differential media. This media has different chemicals in it that provide visible differences between two different types of cell. So the chemical will make a visible difference, allowing you to see differences between two different types of cell. The difference will usually be a color, but it could be something different happening on the plate. The difference will not be related to how well the organism grows on the differential media. But it does make it easy to distinguish colonies of different types of microbes. An example of a differential media is blood agar that allows you to separate the bacteria depending on their hemolytic ability. Let me blow this up. So here we have microbes which are white smeared in the gamma. That's the Greek lower gamma letter. And you can see the blood, red blood cells on the blood agar, come all the way up to the white of the bacteria, meaning these bacteria have no hemolysis, and that's called gamma hemolysis. And these bacteria, which is kind of a smear of an alpha in the off-white color, we notice the red blood cells are red up until here, but around the bacterial growth, we have a darkening of the media, and it's most noticeable inside the alpha, where inside the alpha it's all dark. That darkening, or also sometimes it looks greenish, is from the partial hemolysis of the red blood cells. And partial hemolysis we call alpha hemolysis. Why this is in the alpha, the Greek alphabet alpha, because that's alpha hemolysis. This is gamma hemolysis, and that's a gamma in the Greek letter. Any question about that? And as you can see, let's blow this up a little bit because that alpha hemolysis is pretty small. There is a darkening around the cells from the partial hemolysis of the blood. Now, here we have in the white, the beta, the bacteria, And around the beta, we have a clear zone inside the beta as well. That clearing is from the full hemolysis of the red blood cells. So here we have the red blood cells and they're red up until here. And then we have a darkened ring. You normally see alpha hemolysis around beta hemolysis. And then beta hemolysis is where the red blood cells are totally lysed. You can hold this plate up to a clock or up to the light, and you can actually see through the plate in the clear zone, in the complete hemolysis. I like to look at a clock sometimes and then see the time through the part of the complete hemolysis. Complete hemolysis is called beta hemolysis, and that's why that's a beta Greek letter shown there. Any questions about differential media or blood agar? All right. We also have combined media, selective and differential media. An example of a selective and differential media is eosine methylene blue or EMB plates. It is both selective and differential. It is selective in the sense that gram-positive bacteria are selected against, and then gram-negative can grow on the plate. So it's selective for gram-negatives, selective against gram-positive bacteria. Why it's selective against gram-positive, it has two dyes, eosine and methylene blue, and they get inside the gram-positive cell and then prevent the gram-positive cell from growing. It's also differential on the basis of lactose fermentation. This media has the sugar lactose in it, and if the bacteria can ferment the sugar lactose, the bacteria will turn black or metallic greenish in color. So that's sort of metallic greenish there, and there's black. That's from the change in the pH. When you ferment the sugar lactose, you make an acid, and that will change the look of the cells in acid to black in color or metallic green in color. If the bacteria can grow on the plate, they are gram-negative, but if they cannot ferment the sugar lactose, they will be their normal color, which here would be, it looks like white in color. If the bacteria are off-white normally, then they'll look off-white. So the color will depend on the color of the bacteria. Any question about any of that? So there are selective and differential media, both selective and differential. There is also something we call an enrichment media, and this media encourages the growth of certain microbes to grow and discourages the growth of other microbes. So an enrichment media is selective by its nature, but we don't use it as a selective media. We use it to encourage the growth of certain microbes to grow, and it increases the numbers of the desired microbes to detectable levels so that you can actually, I don't know, get a handle on that and then get it to grow up as a pure culture. Realize the enrichment media will not make the culture pure. It'll just increase the chance that the microorganism you streak out as a pure colony will be a microorganism of interest. An enrichment media is particularly useful if the targeted microorganism is only a small proportion of the total population, such as if you're looking for a microbe from the soil or some other environmental area where there's many different microbes growing, or you're looking for a pathogen in a fecal sample, and you're only looking for one pathogen, and the fecal sample is going to have thousands of other microbes. You're not interested in the other thousands of microbes. You're only interested in the human pathogen. What you can do then is grow the culture, which is a mixed culture, in an enrichment media to enrich for the organism of interest. For example, suppose we want to obtain from the soil a few phenol-degrading bacteria. And in the soil, there are thousands of other bacteria, probably hundreds of thousands of other bacteria. What you can do is take this soil bacteria and inoculate it into a tube, which is an enrichment tube, where the only carbon source is phenol. And so the only organisms that can grow in this tube will be organisms which can degrade phenol. And what you do is let me get it in this here and bring up paint again. So here's your soil. Take your microbes from the soil, put it into a tube, an enrichment tube, and the microorganism of interest might be from the soil, 1 cell out of 1,000,000. Let's see how I can do this. I don't know if I can do 10. Let's just go, I don't know if I can do a superscript here. So let's just go EE6. That's the old way. Actually, it should be capital EE. Up. So 1 out of 1,000,000. And we put it into this enrichment media, which would be something that had phenol in it. And what you do is you increase the cells 100-fold. So it's going to be 1 out of 1. I'm just going to go EE4. I think you understand what I'm talking about. Or 1 out of 100,000. And then we can, oops, wrong thing. Take the cells that grow out of that and put it into another tube, enrichment tube. And let's say it enriches 100-fold. Now we have one, oops, got that wrong. One cell out of 100. Oop. That is enriched enough that if you were to streak out 100 colonies, you would get one colony. That would be your colony of interest. And that is actually something you could do. You could streak out 100 colonies and just test all of them, and you would find your bacteria of interest. Anyways, that's how you use an enrichment media. Any question about that? All right, so this slide shows you the different types of media and then the purpose of it: chemically defined media, a complex media, a reducing media, selective media, differential media, and an enrichment media. And remember, an enrichment media is a selective media, but we don't use it specifically as selective. What we use it for is to enrich for a culture of interest. All right, obtaining pure cultures. A pure culture is a culture that contains only one species. Whenever you're working in microbiology, we usually use pure cultures because if you had a tube that was a mixed culture and you wanted to know, I don't know, if Staphylococcus epidermidis was making the tube yellow, and it was a mixed culture, you wouldn't know which culture in the tube made the tube yellow. So what you do is you grow it up as a pure culture and then put it in the tube, and only Staphylococcus epidermidis is in the tube. And if the tube turns yellow, you know that that is because Staphylococcus epidermidis turned the tube yellow. Any question about any of that? A colony is a population of cells arising from a single cell or one spore or two cells or a clump of cells. So a colony can arise from a group of attached cells. And that's why when you first streak out a plate, I will warn you that about 30% of your colonies are not pure because they are started by more than one cell. And therefore, we say that a colony is called a colony forming unit because it is may be formed from more than one cell. To obtain single colonies, which is a critical part of obtaining pure cultures, we use the streak plate method, also called the streaking for colony isolation. And you have to do it 2 times to ensure you get a pure culture. In fact, even if you do it 2 times, you only ensure that your culture is 95% pure or higher. It will never be 100% pure from colony streaking because in science, we're never 100% sure. There was one time there was an instructor who was trying to get a pure culture, and he didn't know it, but the 2 cells had to be together, 2 different cells. They were both metabolizing. I'm trying to remember what it was. They were metabolizing, I think they were metabolizing a different form of phosphorus. And you needed both of them there because he's only providing one phosphorus source, and both of these organisms did not. How was that possible? I guess he was supplying one phosphorus source, and he was trying to isolate the other organism that did not use that phosphorus source. And so he had to have 2 of the cells together in order to get that organism to grow. Anyways, my point is he was streaking out for colony isolation, and he did it 2 or 3 times, thinking he had a pure culture. But in reality, he had 2 species there because the species of interest could not grow in the media, and so it had to grow in media containing the other bacteria species. Okay, my point is we never are sure you have a pure culture when you're streaking out for colony isolation, because even if you do it 2 times, you can only say that, well, I'm fairly certain that 95% of the time or higher, the colony was formed from one cell. And if you do it only 1 time, you can only say that you're only 70% certain that the culture is pure. And 70% is not good enough in microbiology. Fortunately, 95% of the time is good enough in microbiology, because most of the time when you have a colony that you've streaked out 2 times through colony isolation event, it will be 95% of the time pure. And so 95% of the time you'll have a pure culture, and then 5% of the time you will not. So most of the time you will get a pure culture, which is good enough in microbiology. And it's fortunate in microbiology that we do not need 100% because you would almost never get 100%. As I gave you the example where my instructor friend was trying to grow up this bacteria species and would not grow on the media he was supplying it. So each time he streaked it out, he got two different organisms because the one organism of interest would not grow on the plate. It had to grow in the presence of the other organism. Any question about any of that? All right, let's talk a little bit more about streaking for colony isolation. What you should do is take your loop, put it in a tube, touch the inside of the tube to get the worst of the bacteria off, and then streak it out on a plate. You want to streak it out about 20 times, 20 to 30 times in one sector. This is not showing you 20 to 30 streaks because they're trying to show you the streak. And then you sterilize the loop by putting it in the bacterial incinerator. And then come in sector 2, start streaking, and then go to sector 1, where you have a lower concentration of the cells. You don't want to come into sector 1 where there's a high concentration of the cells, because as you streak out sector 1, this will be the lowest dilution of the cells, and this will be the highest dilution of the cells. You want it where it's lower to pick up some cells. And you should only go into sector 1 about 3 or 4 times. So they've gone too many times, but that's okay. And then streak out sector 2. Come on. Sorry, my mouse has died. There it is. So actually, you should only come about right there and then come down here. And sector 3 isn't quite shown correctly. And what you should do is take your loop, put it in the bacterial incinerator, and then start in sector 3, which I'm not sure why that's not shown. It's probably here, it's just not shown. And then go into sector 2 where it's dilute. And then you should come into sector 2 at least 2 times. So this should come up here 2 or 3 times. And then streak out sector 3. And if you do sector 4, which isn't going to be possible here, I can maybe do it here. Do it one more time. Put your loop in the bacterial incinerator. We don't have much space here for sector 4, but only come down to sector 3 where it's dilute. Go down about 2 or 3 times, and then streak it out for sector 4. Okay, you don't need to do 4 sectors. You may only do 3 sectors. The point is someplace where you're streaking, you will be streaking isolated cells, which will grow up to be isolated colonies. In this case, it happened in sector 3, but it's fine if it happens in sector 1. And if your stock culture is really dilute, you could get it in sector 1, where you're streaking out isolated cells, and then you get isolated colonies. And usually you won't get anything in sector 2 or sector 3. Okay, doesn't matter where the isolated colonies form, you're just doing this colony streaking, so you will get it in one of the locations, one of the sectors. Any question about that? Colony streaking works well when the organism to be isolated is present in large numbers compared to the total microbial population. If the organism of interest is very rare, then you'll need to grow it first in an enrichment media and then streak it out. Any question about any of that? All right. When we're talking about bacterial growth, oops, sorry, it's time for me to end, so let's put it here. Are there any questions? All right, if there's no questions, I'm going to end it here and I'll see you at 7. Let's make it 7:02 for the lab. Okay? All right, I'll see you then. Come on.